Our next speaker is Tian Zhu, and he's going to speak about biofuels in China.
Well, first, I'd like to thank the organizers for giving us this opportunity to talk about our efforts to build a bioenergy institute in Chinese Academy of Science. Everybody recognizes that synthetic biology has such an important application at bioenergy. So I truly believe that this opportunity to speak here belongs to my colleagues back in Qingdao. So I'm going to first give an overview about our efforts there. And second, I would like to talk about my involvement from human microbiome to environmental microbiome. And most of our colleagues this morning, they talk about how to construct a genome. And here, I would like to try to convince everybody that decoding a genome, although it has become more routine in many people's way, but still, this is a front line, and I think it's going to give us explosive growth in the future, and it's going to impact the growth of this new discipline of synthetic biology as well. How do I go next? Sorry. Okay. The right one? This way? Yeah. Okay. Where is the pointer? Pointer? That's it. Thank you.
It's connected to your computer, not this one.
Okay. Well.
You can use that. It's not this.
Okay. Thank you. Okay. Yes. Sorry for that. Well, just to give you an overview about Chinese Academy of Sciences, many of you, I believe, have collaborators in this institute. It is a military-level government branch, and it has 90 research institutes across China. And we are here, and we consider we are at the neck of this big chicken of China. And Beijing is here, so it's heart. And of course, Beijing is important as the heart, but at the neck, we are also important too. And so this is our new institute, which has been completed about 2 weeks ago. And we just moved in last weekend. So this is still called Chinese Academy of Science, Qingdao Institute of Biology and Bioprocess Technology. So why we are working on this? It is a new national research institute in Chinese Academy of Science, and its establishment reflects the three demands from national level to regional level. China, as a nation, it has a huge energy demand, and it also has a huge demand for environmental protection. And Shandong province as the number 2 in GDP, and right behind Guangdong province. However, it is a champion in energy consumption. And of course, this is not good. It has a potential for heavy environmental contamination. And Qingdao as a city is historically heavy industry, international acclaimed coastal tourism area. So it's important for this area to develop bioenergy, and also for China as a nation as well. So this is the size of our institute in 2010. So in our view, bioenergy has 4 key bottlenecks in converting biomass and biowaste into useful materials. One is the biomass supply, the quality and quantity of supply. Second is the efficiency of biomass conversion. Third is the process integration and scaling up issues. Fourth are the scope and quality of new products. So these 4 bottlenecks reflect our research priorities in the institute. So these are the major research areas in our institute. First, cultivation, screening, and engineering of plant species for bioenergy application. Second, to screen and reconstruct functional microbial isolates and communities for bioenergy applications. Third, microbial catalysis and metabolic engineering or biodegradation processes. And fourth, also reaction, separation, scaling up, integration issues in bioprocess engineering. And also biotransformation process and computational simulation. And finally, we also have the policy issue because we carry the mission of advising the government in terms of bioenergy development. So we also have this research area. So I stole this slide from the website of DOE. And I think this pipeline reflects our organization of our research. We have Biology Genome Center, which is responsible for looking at an exploitation of the natural products, plant species, algae species, and microbial species and communities. They are of potential use in biology. We also have this Bioconversion and Catalysis Center, which is responsible for designing enzymes and improving their efficiencies in industrial processes. We also have the Process Engineering Center, which is responsible for designing reactors and essentially how to upscale the molecular process to an industrial process. We also have a Bio-based Product Center, which is mainly to develop new biochemicals and bioenergy molecules. And we finally also have the strategy center. And my colleagues and I are responsible for building up this Bioenergy Genome Center. These are the teams currently that we have in the 4 centers. Our center right now has energy plants, energy algae, and microbial resources, and functional genomics, and bioinformatics engineering team. These 5 teams. 3 of them reflect the research target that we are aiming at, and 2 of them reflect the approach that we are taking. So this is our mission. We want to take a systems biology approach to understand energy, microbes, energy plants, including algae. And these are our key targets. Our major approach, I think all of you are very familiar with these approaches right here, and I'm not going to go into details. Essentially, it is an omics approach. So within the last 3 months, we have assembled quite an interesting, very nice team here. And we have this functional genomics team, which I lead. We also have this Dr. Li leading the microbial resources team. And we also have Dr. Zhou, whom we recruited from University of Georgia to build this energy plants team. And we also have Dr. Liu leading this energy algae team. We also have graduate students and staff. Currently, we have 30 people together. These 5 teams together, we are building these 5 platforms. One is the high-throughput genome sequencing analysis platform. The second is the functional genomics platform. The third is the microbial physiology and culture bank platform. The fourth is the modeling and software development platform. And the fifth is the plant physiology platform. So that was enough overview for what we are doing. But right now, in the next few slides, I'm going to tell you how my research interest evolved from human microbiome to environmental microbiome. We think what we are doing right now is a natural extension of human microbiome because, in fact, in this part, the microbial communities in the nature and human gut and in animal digestive tracts, they are responsible for converting plant biomass into smaller molecules like glucose. So these are the processes that we are investigating in this center. And this is our summary of an overview of my previous research program in the human microbiome side. Essentially, I have been interested in a question, which is the structural function and regulation of polysaccharide degrading microbial communities. I use human gut microbiota as an example. And the colleagues and mentors I work with before are Jeffrey Gordon and Rick Wilson at Washington University Genome Sequencing Center. I have also been developing tools for microbial genome sequencing and functional genomics. And in addressing this scientific question, we focus on 3 different parts. One is the discovery of functional genes and their regulatory mechanisms. And we use Bacteroides theta-iota omicron, which is a dominant symbiote in human intestine, as a model. Second, we model the biological processes, especially of the polysaccharide utilization process in the gut as a model. And we look at how the species interact to form a functional community. Third, we elucidated the evolutionary mechanisms of these symbionts, which are co-evolving in the gut, by looking at the genomes of 3 dominant species in the gut, which is Bacteroides theta-iota omicron, Vagatis, and Dispersonis. In this tool development, I just want to highlight this part, which is a design, validation, optimization of new generation microbial genome sequencing strategies for human microbiome project. I think many of you recognize that NIH has been pumping like $25 million as a pilot project for the ROMAT project, so-called ROMAT project. And this ROMAT project, starting from April of last year, has been focusing on human microbiome. So we in terms of our strategies that we developed for sequencing the metagenomes and individual genomes of this microbial community and its implication for human health have been addressed in this paper, so you can take a look. So let me give you an overview how we approach this human microbiome community. So human microbiome is interesting because it's an extension of human genome project. And we consider human as a superorganism because we have 10 times more bacterial cells in human cells. And gut microbiota contains more bacterial cells than all of our other microbial communities combined. So it also has the highest density of microbes in nature. So we use gut microbiota as an example to study the human microbiome. And in terms of evolution, actually, we not only undergo the macroevolution, for example, how we evolved from chimpanzee to human, we also have a microevolution going on in our intestine, because we have a lifelong association for every human being, from our ancestors to our offsprings. And this is also an evolutionary conserved phenomena found in multicellular organisms, and it's called microevolution only on the microbial side as well. And its role in human host physiology has been very, very important. It's involved in colonization resistance, which was discovered 100 years ago. It's also important for digestion observation. And some of the recent observations and findings are listed here. For example, it has involved in response to epithelial cell injury. It's also responsible for biosynthesis of certain vitamins, and it's also important for biotransformation of xenobiotics. It is also a modern system to study ecogenomics in natural complex microbial communities. So these are the questions that we are interested at addressing. So why we pick polysaccharide utilization as the model process to study this ecosystem? First, plant polysaccharides are the only abundant carbon source in digital small intestine and colon. The human cannot make use of them due to the lack of most enzymes for this role, but our microbial partners could help us. And they help us by fermenting the plant-based polysaccharides into fatty acids, short-chain fatty acids, and those provide 10 to 15% of daily calorie intake to us. And of course, this is depending on our diet, individual diet. And actually, some of the recent findings address this issue, that the difference in the composition of intestinal microbiota actually influences energy balance of individual hosts. So our composition of these partners in our intestine have implications for various phenotypes like obesity and more recently diabetes. So this complex microbial community is composed of, based on this study by Edberg and Stanford, they found that three Caucasian adults, based on their 16S sequences collected from them, from the intestine, they found 9 major phylogenetic lineages constituting of hundreds of phylotypes, about 400 of them. And 99% of those phylotypes belong to Bacteroides and Firmicutes. These are the 2 major bacterial phylums in the bacterial domain, about half and half division. And why we are looking at specifically at Bacteroides, because it has ecological impact. It has a broad geographic and ecological distribution, and it is a numerically dominant group in many marine regions and digestive tract of many mammals. And the most important is that they have exceptional ability to degrade polysaccharides, including cellulose. So what kind of role do they play in our intestine for polysaccharide degradation? We first sequenced this genome of the Bacteroides theta alta omicron, and we found it has the most abundant glycoside found in single bacteria, pointing to the molecular basis of one crucial step in the bioenergy conversion in our intestine. Second, we also discovered some of the unique regulators and regulatory mechanisms of polysaccharide degrading loci in this organism. We identified ECF sigma factor. I think many of you are very familiar with this type of regulators. It provides the organism with a grading capacity. And second, we also, for the first time, identified some novel hybrid two-component systems in this organism. And we know two-component systems have a histone kinase and response regulator. And typically, it is these 2 parts in bacterial genome. But here, in this organism, we identify 31 of them. And these 31 of them, each of these molecules has all of the 4 key domains in a two-component systems together in a single polypeptide. So this is probably an important part for the synthetic biology for xenotransduction as well. So beyond that, we want to look into how these communities interact. We only look at one genome, but we want to extend such kind of analysis to many more species. The question we are interested at are, what are the shared and distinct features in the glycobiomes of different lineages? And how do the different functional components on the polysaccharide food chain interact? Third, how do the intestinal environment shape the evolution of the various lineages? So right now, we just use an example. We look at the comparison of 2 species. One is the Bacteroides theta omicron, and it is a gram-negative one. and it is dominant in adult intestine and belonging to the Bacteroidetes phylum. The second we look at belongs to the Firmicutes phylum, the other half of this Firmicutes, the other half of this microbial community. And it is a gram-positive one. It is minor in adults, but it is a major component of the newborn microbiome. And it is a commonly used probiotic for adults as well. So first we compare the scope and the range of scope of their polysaccharide utilization enzymes. And we do see Bacteroidetes delta omicron has a much larger representation, a much wider representation of this type of enzymes. Second, we also look at their loci on their genomes response for polysaccharide utilization. First, let's look at the structural enzymes. These are the glycosyl hydrolases encoded in those loci in the two species. We just in the previous slide we showed that Bacteroides delta omicron has a wider range of polysaccharide utilization enzymes, and for certain type of glycoside bonds, only those enzymes are only encoded in this genome, but not in this genome, which suggests that B zeta can degrade a much more wider range of polysaccharides than B longan. Second, we also look at the other module of this polysaccharide loci. This is SUCC. It has been demonstrated to be involved in the binding of starch, a type of polysaccharide. And these EFG ABC transporters, they are not able to transport polysaccharides. They are only able to transport and bind oligosaccharides, and typically disaccharides. And third, we also look at a regulatory mechanism. We found that in B zeta delta omicron, the dominant type of regulation for this polysaccharide utilization loci are so-called ECF sigma factors. We all know that ECF sigma factor is a type of positive regulators. So in response to environmental stimuli, the ECF sigma factors, which is associated with the anti-sigma factor, which is located in the membrane of the bacteria, will be released and activate the transcription. And in B longan, actually, this is a LEC-I type of repressor. It represses the regulation. And the substrate specificity and the location and the regulatory mechanisms of the two types of regulators suggest that only B zeta can respond to the environmental stimuli from polysaccharide. So what are these telling us? They told us two things. First, there is a hierarchical multi-component polysaccharide utilization process in human intestinal microbiota. Not everybody plays the same role. Second, B zeta specifically, its ability to grade stabilizes the food web and promotes the long-term robustness of the community and the ecosystem. So what I have just shown you is how we kind of taking the genomics approach, look at individual components of this polysaccharide utilization community, and then glimpse into and look into how the efficiency of this ecosystem and the productivity of this ecosystem is maintained. We also look at the time angle to look at how this evolution kind of shaped the genome of these intestinal bacteria. I'm not going to go into too much details here, but essentially we sequence the genomes and look at related species of B zeta delta omicron, and we found that several functional categories are enriched among natural transfer genes. For example, those glycosyltransferases and those involved in CPS, capsular polysaccharide synthesis loci, and also those DNA methylation enzymes, those are involved in restriction modification of genes. So the summary of this history, of this evolutionary part is that there are three processes that drive the Bacteroides' adaptation to a digital card. One is the natural gene transfer. Second is mobile elements. Third is gene application, which I didn't show data for. And these three processes affect the ability of gut Bacteroides to vary their cell surfaces, sense their environment, and harvest nutrient resources in the digital intestine. So I just showed you that we sequenced individual bacteria, and NSF gave us $3 million to sequence 5 over 3 years. But after 2006, everything has changed. Right now, we have 454, and many of you are very familiar. And one thing, this is behind the army of 3730s in Washington University Genome Sequencing Center. Now, a single 454, the throughput is equal to 150 3730. So these have completely changed the scope of our work. So right now, NIH has given us another $3 million to sequence 120 bacterial species. And this is going to be even more in the near future. And just give you an overview about what we are doing here. So these are the 3 platforms currently available. And for example, the solid Solesa, they give you much shorter reads, and 454 give you longer reads. But overall, they all give you much better throughput. So these have completely changed how we sequence microbial community. And you can see more details about human microbiome project from NIH website. Essentially, these are the questions they are going to address. So our previous approach really opens the domain for looking further into other locations of the human body. And so 3 months ago, I moved from Washington University to Chinese Academy of Science to help establishing this biology genome center. And there, what we are looking at is those micro communities. They are of interest in biology application, especially those helpful for cellulosic ethanol production. And I just want to give you an overview about what we are doing here. So previously, this is the current way of developing producing ethanol from cellulosic plants. And this is where we like to go. This is called consolidated bioprocessing. And it has the overall process cost reduced 2 times in theory. And it requires industrial microbes that can degrade cellulose and produce ethanol simultaneously under some aerobic condition. And the key point is that in this CBP, we have to merge these 4 steps together. So we have decided to look at a mixed culture of Clostridium and Aerobacter for this purpose. And why? Because they are able to, some of these are able to directly degrade cellulose, and some of them are able to ferment a variety of monosaccharides into ethanol. And thermophilic anaerobes, as compared to yeast, E. coli, and Xymomonas mobilis, has important advantage because it lowers the cost of producing ethanol. However, these are the challenges. How do we do that? So this is the approach that we are doing. We don't have a lot of preliminary data yet, but this is what we are heading for. We want to take a V phenotype and then through the genotype get to our narrow down to an interesting phenotype. We need to break 2 barriers. One by single cell genomics by breaking these not yet culturable cells, so that we can culture them. Second is that we want to use metagenomics approach to break the barriers of insufficient sampling of natural diversity of genes, and through these 3 steps and get to target genes. So this is the one important research domain in our center to do this. And to do that, we have to sequence their genomes first. So these are the approach that we are doing. And after we discover some important isolates, what we are doing is to understand and improve those microbes for bioenergy applications. And this just shows you the cell, in this case Clostridia, in this case Aerobacter cell, the pathway that converts xylose and glucose into ethanol. And so there are some side products here. So the goal is to improve the metabolic flux in this route and reduce the flux in the other 3 routes. So these are the questions that we are interested in, and we are addressing these 4 questions with omics approach. So these are the 3 technology barriers that we have to break. One is the better method for mutant generation, and second is the more economical, faster method for genotyping mutants. And actually, this one turned out to be the easiest one because after we have the new type of DNA sequencer, we can already break this barrier. But these T1 and T3 end up to be much more difficult. T3 is the optimization of multiple gene controlling phenotypes through genetic engineering. And these are the overall research scheme that we are heading towards. We start with a starting collection of microbes. In this case, we have 31 Aerobacter isolates. We use comparative genomics to understand the genotype and their alterations. We also use mutagenesis to get the mutants and use high-throughput mutation mapping to get back, also to aid the understanding of genotype and their alterations. And we also use functional genomics approach to characterize the mutants and the wild types. And by integrating these 2 types of data, we hope to understand the mechanisms underlying these ethanol-producing phenotypes and other important phenotypes like inhibition by ethanols and other inhibitors. So sorry that I'm taking a little bit late. With this, I just want to give you thank Zhou, the lab in University of Oklahoma. We work closely together at the thermoaerobatic genetic engineering part. I also want to thank my previous mentor, Jeffrey Gordon, who has led me into the human microbiome site. And I also thank these members of my group. And our funding are supported by Chinese Academy of Science and National Science Foundation of China. And these are the websites. And when you have time, please visit us. We are located at Qingdao. Thank you.
Thank you very much. Are there any questions? Yes, Rob.
Just curious if you're thinking about streaming all of those metabolic pathways into something other than ethanol, given that other fuels might be, since you're starting from scratch, given that other fuels might have better physical characteristics.
Oh, absolutely, that's a great question. And we, for example, butanol, and there are some other molecules that are under development. But right now, the strains that we have can only produce ethanol. They cannot produce butanol. So this kind of underscores the limits, the barriers in the availability of natural resources. For example, right now there's a very limited type of natural microbes that can produce butanol. Of course, there have been a lot of excellent research going into how to modify E. coli to produce butanol. But right now, we haven't gone into that. Thank you.
I had a question. I might have missed this. So in your new center, are you going to focus on a different microbiome community? Are you going to still be doing gut bacteria for your
Well, I tend to think that the gut bacteria is a bioenergy application, but most of people don't agree so. They actually, we want to, our priority is on the rumen bacteria, for example, those that can degrade cellulose. Mm-hmm. Human gut cannot degrade cellulose.
So you're going to be focusing.
Yeah, we are mainly focusing on environmental microbiome of interest to biology. Yes, especially cellulose degradation.
Right, but from the gut of the cow.
Right, rumen, yes.
Okay. Any other questions? Okay, thank you.
Thank you.